So many thanks to Laura and Paul for this terrific symposium, and also for keeping us living longer by this cold room. It's great. So I would like to maybe put things in a slightly different perspective. I don't want to discount anything that's gone before. I think senescent cells are a driver of aging. So this is the idea. Oh, this is a disclaimer. I am a founder of this company. I'm not going to talk about any company stuff. We all know that aging is basically major cause for all these terrible diseases. And of course, as you heard yesterday and also today, we don't think this is a coincidence. We think there are aging processes that are driving these very, very different types of diseases. And there is now mounting evidence that cellular senescence is a driver, one of the drivers of this myriad diseases that we associate with aging. So the way we've evolved, so I should say the field has evolved enormously in the last several decades. And we tend now to think of senescence as a stress response. So Felipe, where's Felipe? You can put it in the middle of his slide.
I'm here,
I can't see you. Okay. But and as other people have shown and as Jim has shown, it has this bipartite phenotype. We haven't talked much about this growth arrest, but I want to emphasize there's 20 years of data that indicate that this growth arrest is crucial for preventing cancer. So the thing we don't want to do is prevent cells from undergoing senescence. We know what happens. Animals and people die a terrible death due to cancer. So what we want to do is, though, is to be able to do something about this secretory phenotype that Jim alluded to, which is multifactorial and contains lots and lots of components, many of which can be deleterious, not all of which. So I want to talk about the types of stressors that will drive cells into senescence and contrast them. So this is a partial list, again, a partial list of the things that will cause cells to adopt a senescent phenotype. And it ranges from things that damage the genome to oncogenes, things that derange metabolism, things that stress other organelles in the cell. And I guess we can call these sort of physiological or non-physiological stressors. But recently, in the last year or so, there have been 2 labs that have identified senescence in very beneficial contexts. So one is during embryogenesis. This is the lab of Manuel Serrano and Bill Keyes. And what they've shown is that certain structures within the developing embryo, and this is true for mouse and human, are fine-tuned by the presence of senescent cells, which seem, again, through the secretory phenotype, to secrete factors that then help fine-tune morphogenesis during development. And then I'll show you data from our lab, but there are at least several other labs now that have shown that the secretory phenotype can also participate in beneficial tissue remodeling, such as wound healing. So there's going to be a balance, and I think this is really going to be a refrain that I'm going to come back to at the end, that understanding senescence really requires, first of all, that we understand the evolutionary context in which this stress response evolved, and then being able to balance good things versus bad things. If any of you listen to Science Friday, you know exactly what I'm talking about. So a quick word about senescence markers. Many, many labs have worked on this problem, and over the years there have been lots of markers that have been identified that can identify senescent cells in vivo and culture. There are no senescence-specific markers of which I am aware, and so we need to use multiple markers. But I want to highlight a few. The first is the secretory phenotype, which I'll come back to. And I want to make the point that we can now bin the secretory phenotype into 2 main classes based on their molecular control. And the first is this mixture of cytokines, chemokines, the things that Jim talked about, as well as I'll show you some bioactive lipids. And what sets this program apart from this one, which I'll tell you about in a moment, is that this process is completely p53 independent. Contrast that with the secretion of what is now a growing family of proteins and small molecules, which are now called DAMs, damage-associated molecular patterns. They used to be called alarmins, but now I think everyone calls them DAMs. And we identified the first such DAM that was secreted by senescent cells, but now we have evidence that there's a number of them. This process is completely p53 dependent. What all of these secretory and the other thing, of course, is the expression of p16, which is the basis for our transgenic mouse models, and the fact that the cells resist dying. Let me show you a little bit of data. This is a protein called HMGB1. It's primarily a nuclear protein. It's a chromatin remodeler. And it is completely absent from the nucleus of senescent cells. And instead, it is secreted into the extracellular milieu. This is the first alarmin that has ever been identified, and as I said, we now know there are several other alarmins or DAMs that are secreted by senescent cells. This is the secretory phenotype. This is an antibody array. And what this array shows is that there are lots of things that are secreted by senescent cells that are not secreted by non-senescent cells. These are 5 different types of human fibroblasts. And as I said, this is completely independent of p53, whereas this process has nothing to do with transcription. So again, doing RNA profiling is useful, but in the end, we do need to look at proteins. This is new data from Chris Wiley in the lab showing genes that code for intermediates of lipid metabolism, especially proteins that, genes that give rise to the biosynthesis of both prostaglandins and leukotrienes. And senescent cells now we know are a very rich source of both types of bioactive lipids. What all of these secretory components have in common is they all have the potential to drive inflammation, primarily infiltration of innate immune cells. And we've shown that in a number of ways. And as you know, inflammation is one of the major underlying causes or exacerbators of all of those age-related diseases that I showed you on the first slide, as well as virtually all of the major diseases that have been described with this syndrome. Inflammation, of course, can not only destroy tissue, but it can upset stem cell functions, and it can, of course, promote cancer. So this is ironic, this tumor suppressive mechanism that in the end is driving a secretory phenotype that can be pro-cancer. So the model then that we've been working on for a number of years is that with age we accumulate senescent cells. That's been demonstrated in virtually every vertebrate species that I'm aware of. And these senescent cells then secrete molecules that can cause neighboring cells to fail to function, and that would drive the degenerative diseases of aging. And we've also shown, we and others have shown, that the secretory phenotype can also convert pre-malignant cells into malignant cells. And so in the end, both the hyperproliferative as well as hypoproliferative diseases can be driven by these, the secretory phenotype of senescent cells. So of course this could be very depressing. You can't live without senescence, but if you live with it, you're going to die anyway. So what do we do about it? As Jim discussed, there are a number of pathways that drive the secretory phenotype. Those pathways have target proteins. Many of those target proteins have drugs already in the clinic, and we've done a lot of work on that. It's not our favorite method for alleviating the deleterious effects of senescent cells because you need constant pressure from the drugs. So these are not senolytics. These are drugs that inhibit pathways that drive the secretory phenotype. So the drugs need to be constantly present. As soon as you take them away, the cells start secreting again. And many of these pathways are crucial pathways for optimal function. For example, the DNA damage response pathway is one of the pathways that drives the secretory phenotype. So this is why we and others have favored the idea of eliminating senescent cells, and we can now do that. Jim talked about Jan Derson's mouse model, which is the same principle as our mouse, which I'll just explain in a little bit more detail. As Jim mentioned, we use a large bacterial artificial chromosome containing the endogenous p16-ink4A locus of the mouse, and we clone downstream of the p16 promoter a fusion protein that enables us to visualize senescent cells with luciferase. This is ranilla luciferase, sort them through an RFP, then kill them with this drug, ganciclovir, which is acted upon by this viral thymidine kinase, it produces a phosphorylated derivative that is a very potent DNA chain terminator. Now, senescent cells don't replicate nuclear DNA, so it's not acting on the nuclear DNA, but it does act on mitochondrial DNA, completely fragments it, and causes the cells to die by apoptosis. So this is from Jan van Dersen's latest paper, in which he took middle-aged mouse, so this is now his transgene, same principle, different drug, different killing mechanism. He took middle-aged mice and then treated them intermittently with this, the drug that causes the death of senescent cells, and asked, are the animals any better off? And the answer is their health span was remarkably improved. So there were many indicators of health that were improved, and median lifespan is consistently improved. I just want to draw your attention, though, to maximum lifespan. So even though we do believe senescent cells are drivers of disease and age-related degeneration, we still have to invoke either something else or the fact that these cells are only partially responsible for setting maximum lifespan. Okay, so we have used our transgenic mouse now in a number of settings, and I'm going to very quickly show you what some of those settings are, how senescent cells can be deleterious, and how eliminating those cells can be helpful, at least to the health of a mouse, and then tell you about how we're thinking about how we can place these deleterious effects in the context of evolution. So the first thing has to do with DNA-damaging chemotherapies. As many of you know, many chemotherapies are genotoxic. They tend to drive cells into senescence, and there's now a rising problem in the clinic, especially of children who were treated for leukemia or lymphoma with these DNA-damaging agents. And these kids, who are now middle-aged adults, are showing up in the clinic, and they look 20 years older than they are. So we hypothesize that maybe some of the deleterious effects of chemotherapy could be due to the accumulation of senescent cells. And this work was done mostly by Marco Di Maria. So here's an example of an experiment Marco did. He treated our mice, so we're now looking at renylluciferase reporting the presence of senescent cells after a single dose of these different chemotherapeutic agents. And what is remarkable about this luminescence is that it persists. So months, even a year later, the cells have now a burden of senescent cells that exceeds an age-matched control. We can eliminate that luminescence as well as markers of senescent cells by treating the animals with ganciclovir. It's just 5 days of ganciclovir treatment. So now we can ask, so what? So you have a burden of senescent cells. Why is this bad? And it's bad for at least 3 major problems that have been identified from survivors, cancer survivors who have experienced chemotherapy. And the first has to do with metastases. So this is now a model in which we've taken breast cancer cells that are now expressing firefly luciferase. So now we're going to follow tumor cells in vivo by luminescence, but it's a different luminescence. It's not Renilla, which is our transgene. So it's well known if you take these breast cancer cells, you inject them into the mammary gland, they rapidly metastasize to the lung and the liver. So now we can do the following experiment. We give the animals breast cancer, we wait a few days for the tumors to develop, and then we give them chemotherapy. And then we either clear senescent cells or not. And we ask what happens to the primary tumor and what happens to the metastases. So here's the primary tumor, here's a metastasis in the liver, and here's a metastasis in the lung. If we treat these animals with ganciclovir after the chemotherapy, we have variable effects on the primary tumor. It's totally inconsistent. It seems to be stochastic from animal to animal. But in all cases, we completely eliminate the ability of those cells to metastasize to the lung and the liver. We interpret this to mean that we change the systemic or local milieu of those tissues, which is being driven by the senescence-associated secretory phenotype. We haven't proven that definitively yet, but that's our working hypothesis. Now, the second problem with chemotherapy is it's often limited by cardiotoxicity. In fact, often the amount of chemotherapy that a physician can give a patient is limited by the cardiotoxicity, and we can measure this in the mouse. So we can take a mouse and give it chemotherapy, in this case doxorubicin, which is an anthracycline. Those are the worst drugs for cardiotoxicity. And we can show that if we give the animals the chemotherapy and wait 3 weeks, by that time the animals have already shown a reduced heart function, and that's shown by a decline in ejection fraction and a decline in fractional shortening. If after the chemo, a few days after the chemo, we eliminate senescent cells with ganciclovir, we can prevent that cardiotoxicity. If we wait too long until after the heart defects have developed, we can't rescue. So this is preventing the cardiotoxicity, not necessarily rescuing the heart from the cardiotoxicity. And the last example was something that Felipe mentioned yesterday, which is chemofatigue. We can model this in the mouse by putting them in a metabolic cage. And one of the nice things about the metabolic cage is it has a running wheel, so we can measure activity. This turned out to be the most useful measure of the metabolic cage for our purposes. So mice are nocturnal, and at night they will run kilometers if you let them. And so now we can ask, how active are these mice? So here are non-treated mice. They spend about 40% of their time on the running wheel. If we give them chemotherapy, that drops down to less than 10%. But if we now eliminate senescent cells again within a few days after the chemo, now these animals are as active as the non-treated animals. So we're cautiously optimistic that the idea of using senolytic agents might now become part of a chemotherapy regimen to help alleviate some of the side effects of chemo. Now, I'll very, very quickly talk to you about 2 other diseases, very different types of diseases related to age. One is Parkinson's disease. This is a collaboration with Julie Anderson's lab. And she was inspired to come to us because she did this experiment. This is the substantia nigra from autopsy material from humans that were either had Parkinson's disease or age matched. And there was this increase in this marker of senescence called P16. And when we looked at some of the SAS markers, they were also elevated in the Parkinsonian's brain. So we can give a mouse Parkinson's disease by treating them with this herbicide, paraquat. It's known from human epidemiological studies that paraquat causes Parkinsonian symptoms in people. That's also true in